Very good evening, everyone. Please confirm if the screen is visible. Please confirm if my screen is visible and is my voice loud and clear? Very good evening, Aditi. Very good evening, Sheikh. How are you all? Very good evening, Vidya, Bhaskar, Chitra. So yes, are you all excited for the another session? Very good evening, Kartika. Thank you all for confirming. Very good evening, Parveen. So I'm Dr. Tanushree and I welcome you all to another session of CUET PG Prarambh Series. So today in this session, you will know about principles and applications of chromatography and spectroscopy. So why these two techniques? Why you should study these two techniques? First reason, it's quite obvious that it's given in your syllabus, right? It's given in section 1 of your syllabus, that is techniques. And it is very important, these two topics are very important from exam point of view because you can expect questions from these two techniques. So this is the first reason that we are studying today about these two techniques. This is the reason I have selected these two techniques for today. Then second reason is that I'm sure you all aspire to you all want to make a career in field of life sciences, right? And when it comes to life sciences, any field of life sciences, these two techniques are very, very important. They are basic techniques, okay? And therefore, it is very, very much important that you should know about these two techniques thoroughly. Now, another question is that how you need to prepare for these, like how you need to prepare for the exam. Like if you're preparing these two techniques, what you should study. It's just that you have to open the book and start reading line by line, or you just open the notes and start reading it. No, that is not the good approach. Okay, that is not the good approach. The right approach is that you have to emphasize on the concept, especially from where the questions can be asked. Because these two techniques are very much in detail, right? Like if you start studying about these techniques, they are very detailed. So it's very important that you should understand what are the concepts you have to emphasize. Okay. And this is the reason BioTecNika have launched this CUET PG Prarambh series to help you prepare better for the exam. Because when it comes to exam like CUET entrance exam, which is MCQ based, objective type. So in that case, you have to specially emphasize on the concepts from which the questions can be asked. So today in this session, you will learn some concepts which are very important from exam point of view, right? And also you will understand this topic better. So stay tuned, stay with me till the end, because in the end a feedback link will be shared with you all. You have to fill the form, and even the notes of these two techniques will be shared with you all. Okay. So let's get started. I have divided this session into two parts. First is the chromatography. Okay, the first session you will know about chromatography, and another session you will know about spectroscopy. Right? So, what is chromatography? Yes, what is chromatography? Like if I break this word, just a minute. If I break this word, right? So, chromatography, right? So, it means to write with colors, which is literally translated from Greek roots where chroma means color and graphene means to write. And this is the reason I'm saying that it means to write with colors. And this technique, it was first used in 1903 by Russian botanist Mikhail Sweat. And he used the term chromatography when he was separating the plant leaf pigments on solid adsorbent. So why he used the term chromatography? Because he observed color bands on the solid adsorbents. And these color bands are being produced because of the separation of the pigments present in the mixture on the solid support, on the solid adsorbent. And since then, this technique has been widely used. It has become an invaluable tool for separation and identification of compounds. So IUPAC, that is International Union of Pure and Applied Chemistry, has also defined chemistry that it's a physical method of separation, where the components to be separated are actually distributed between two phases, where one is the stationary phase and one is the mobile phase. So I'll discuss about stationary phase, mobile phase, analyte all in detail, step by step. But the question arises that why you need this technique? Have you ever think about it? Have you ever thought about it? Why you need this particular technique, chromatography? Just imagine if you don't have spectroscopy and chromatography technique, what will happen? Why we need this chromatography technique? So the basis, the key is separation. Chromatography is very, very much important for separation. Let's consider a chemical reaction. Let's say you have a reactant A which react with B and form a colored product C. Okay, form a product C. Okay. Now, at the end of the chemical reaction, there is a possibility you will have some unreactive A, unreactive B. Some intermediates might have been formed during this reaction, that will also be there. So if we want to analyze C, if we want only C, don't you think you have to separate it from other unnecessary or unwanted components, right? So this is the reason we have to use chromatography. So the key here is separation, and separation of chemical component is vital in any type of chemical analysis. So if I ask you what is the importance of separation in the laboratory or in any industry, so it is vital for any type of chemical analysis. Separation is vital for any type of chemical analysis. Now, I have told you two things till now, right? Like what is actually meaning of chromatography and why we use chromatography. So I just want to highlight one point here, students, dear participants, that when you're preparing for any topic, be it a technique or be it any topic related to biochemistry, molecular biology, don't start it just like that. Try to understand, ask yourself why I'm reading it. Ask yourself what is the importance of this technique. Imagine what will happen if this technique is not there, or a process of respiration is not there, or glycolysis is not there, or any other cycle, light reaction is not there in photosynthesis, right? So in BioTecNika, we specially focus on the basics. We always start with basics, and we always emphasize our students to learn something which is out, something new, something out from the book, and we help you understand the concept better and make you prepare well for the exam. So not only you not only uh, can be a part of BioTecNika by attending this session, but if you want to connect us, you want to remain connected with us, you can join our Telegram, okay? Or you can download the BioTecNika app, where you will be where we you will get updates on about the certification courses, or you can go through the podcast, you can enroll yourself to certification courses, attend the webinars, and even can get updated about the recent jobs which are there. So here is the link for the Telegram, right? And Telegram group, and this is the app you can download. So coming back to our chromatography topic again, you have to understand that in every chromatography there are three main components. So just learn it systematically. Okay, don't jump to the application first. We go step by step. First we understood what is chromatography, why we use it. Now what are the main components of chromatography? So there are three main components that you have to remember. One is the stationary phase. Now from the name only you can make out stationary, that is something which is fixed, which is not movable, right? So this stationary phase, it can be like it's a simple liquid which you can absorb on some solid support. Like for example, in paper chromatography, the stationary phase is water which is adsorbed on the cellulose paper, right? So stationary phase is composed of a solid phase or a layer of liquid adsorbed on surface of a solid support. However, mobile phase, it is something which is mobile, right? So this phase is mobile, and it composed of liquid or gaseous component. And last part is analyte, which is actually the substance which has to be separated during the chromatography. So these are the three main components that you always have to remember, right? Now coming to principle. What is the principle of chromatography? So always remember that basis of all forms of the chromatography is distribution or partition coefficient. So you have to be familiar with this term, okay? Distribution or partition coefficient. So what is distribution or partition coefficient? You have a sample, right? Now that sample will have mixture of components, substances that you want to separate. So here I have taken the example of paper chromatography. So where my stationary phase, like here I have taken a support that is paper, cellulose paper, on which I will apply my sample, right? Which is comprising of mixture of the components which I have to separate. Now, as I mentioned in the previous slide, that there are two main components of chromatography, three main components. Two are stationary and mobile, and third one is analyte. So what happens when you provide the stationary phase and mobile phase? So when the mixture is being applied on this paper, and the stationary phase in paper chromatography is water, and the mobile phase, it's a mixture of organic solvents that we use. So what happens? The components, they get separated. How they are separated? Based on how they are distributed between these two phases, that is mobile and stationary phase. So usually partition coefficient is always been expressed. It is always been written in this with this expression. It has been denoted with this expression, that it is concentration in phase A by concentration in phase B. That is your molecule concentration in phase A by concentration in phase B. So this is the partition coefficient, and it is basis for all form of chromatography that you have to remember. Now, chromatography, as I mentioned in the very first slide, that it's a very broad topic, right? So far I have just told you the basics. Now, let's talk about different chromatography technique. So I have summarized the different type of chromatography techniques in this in a flow chart, where you can see that we can classify the chromatography technique on the basis of the device that we use. So device here is the stationary bed that we are using. So either it can be column or it can be planar. So column chromatography, where the stationary phase setup is in a tube. Like here you can see this is a column. Now it can be made up of glass. When you talk about gas chromatography or high performance liquid chromatography, it can be made up of stainless steel also. Planar chromatography is where the stationary bed is shape is planar, like in paper chromatography, in thin layer chromatography. Then, depending upon what mobile phase physical state, again we have classified the chromatography, that is gas and liquid. So, gas, that is your mobile phase, can be a gas or it can be in liquid state. And if you are using a stationary phase liquid, then it is called as gas liquid chromatography. If you are using stationary phase solid, then it is gas solid chromatography. However, if your mobile phase is liquid, but if you are using stationary phase that is liquid, then it is liquid liquid chromatography. Or if you are using stationary phase solid, then it is called as liquid solid chromatography. Then we can further classify the chromatography technique on the basis of mechanism of separation. So we have different chromatography technique, as you can see here: partition chromatography, adsorption chromatography, ion exchange, size exclusion, affinity chromatography. So these are all based on these are the different chromatography that are all based on mechanism of the separation. So in this session, you will be you will be studying about some very important chromatography techniques. So let's first know about partition chromatography. So partition chromatography, where the separation is based on partition coefficient, right? It is based on partitioning of components of mixture between stationary and mobile phase. And it is subdivided into liquid liquid chromatography and bonded phase liquid chromatography, right? So have you ever heard about liquid liquid and bonded phase? Are you familiar with these two type of these partition chromatography subdivisions? Yes, can I get some yes or no in the chat? Have you heard about these two chromatography? Yes or no? So see, basically when we talk about liquid chromatography, here the liquid stationary phase it attached to the supporting matrix by means of physical means. Okay. But when we talk about bonded phase liquid chromatography, here the stationary phase is covalently being attached to the matrix. Like for example, if you have a silica gel, so silica gel in which you have octadecyl side chains which are covalently being bonded to free OH groups. Like here you can see the diagram, right? We have a silica particles and in which octadecyl side chains can covalently bond it to free OH groups. So you can write it as C18. Okay, no. Some are saying no, some are saying yes. So Nutan, I think you can understand well that this is how the stationary phase. This is what we use in bonded phase liquid chromatography. And to others, Aditi and Bhaskar and Vidya, I hope it is clear what is bonded phase and liquid chromatography. And to others who are attending this session. So paper chromatography is a partition chromatography, right? And why I'm emphasizing on paper chromatography is because it's a very basic technique, right? Or it is usually nowadays used for teaching purposes, but it has played a very indispensable role in biochemical analysis because it has the ability to efficiently separate small molecules. Like when you talk about peptides, right? Amino acids, oligopeptides, nucleotides, oligonucleotides, right? So how paper chromatography works? So you have a sample, right? So what you're going to do is you will first prepare a solution, right? Let's say you want to separate plant leaf pigments, so you will prepare a leaf extract, right? And you will apply the sample on this strip of filter paper, right? It is made up of cellulose, and you will apply the sample 2 centimeter above the end, right? So you can see we have left some space over here. So 2 centimeter above the end, you will apply. You will allow the sample. So few drops of sample you will apply, okay? And then you will allow it to dry. Then finally, you will place this paper into this chamber where you have added the mixture of organic and aqueous solvent, right? So you can see the tip of the paper is only being touching. It is being dipped into your mixture of aqueous and organic solvent. Now, what happens in paper chromatography? Your stationary phase is water, and mobile phase is the mixture of water, butanol, acetic acid, or you can use water, tertiary amyl alcohol, or pyrimidine. Okay, so they have a specific ratio that you have to use, right? Now, what happens because the paper has a fibrous nature? The water will be soaked in because of the soaked by the paper because of fibrous nature, and it will form a stationary phase. And the organic solvent, it will migrate as a mobile phase. Now in your sample we have the components, some will be polar, some will be non-polar, right? So based on how they are interacting with stationary and mobile phase, how they are distributed in stationary and mobile phase, they will be separated. So always remember, non-polar molecules will move faster than polar ones. Why? Because here your stationary phase is polar. So obviously polar ones will be interacting more with the stationary phase. The components which are in your mixture, they are polar, they will interact more with the stationary phase. So they will move slowly. But non-polar molecules, as they will be more in mobile phase, distributed more in mobile phase, they will be separated faster. And then if your components that are separated are colored, they can be, you can see them visually. But if they are non-colored, then either you can use some other mechanism like radioactivity or fluorescence, or you can use a solution that when you spray it on the paper, it can react with your component to be steady and produce a colored pigment, so that can be visible, right? So depending upon your aim of study, depending upon your type of mixture that you want, the components in the mixture you want to separate, you can perform this chromatography. And we have different type of paper chromatography also, ascending, descending, right? And here please note that once your components in the mixture are separated, you can find out the retention factor. Okay, so retention factor, it is actually the distance moved by component and distance moved by the solvent. So why this retention factor is important? So it's a quantitative parameter, okay, and it is calculated for every individual component. So this is important because every component has a specific retention factor. So when, let's say, you are separating the coloring agents that are used in food, so every coloring agent or dye that is being used in food sample has a specific retention factor. So if you are analyzing a sample, you get a chromatogram and you can compare it with the standard chromatogram, and can find out what that drug is by comparing their retention factor. Then partition chromatography has two forms: normal phase and reverse phase. Now this is very, very important. Participants, please focus on this information. Normal phase is where your stationary phase is polar and mobile phase is non-polar. You can see the examples on the screen, right? Polar is alkylamine bonded to silica, non-polar is hexane, heptane, ethyl acetate. Reverse phase is just opposite. Here, your stationary phase is non-polar, but your mobile phase is actually polar. Now, why I'm actually emphasizing on this point is because you can directly expect a question on this particular concept. So you have to emphasize on this concept that what is the stationary and mobile phase in normal phase and in reverse phase liquid chromatography. And even I have mentioned here a series of the solvents based on their polarity. So water is highly polar and is less polar. So in order of increasing polarity, even you can refer this series, you can go through it, okay, you can learn this series as well. So let me show you the question which can be asked. In reverse phase liquid chromatography, stationary phase is what? It's a very direct question. You can easily score on this question, and it has been asked. If you go through the previous year question papers, the CUET papers, there such type of questions are mentioned. So just go through it. In reverse phase liquid chromatography, just now I told you about it. So can you quickly tell me what is the answer? Let me see how many of you have paid attention in the class. Yes, can you tell me the answer? In reverse phase liquid chromatography, stationary phase, is it polar? Is it non-polar? See, if you learn about normal phase, for reverse phase, automatically you can recall, right? Or vice versa. So only one you have to learn. Either you learn the normal phase or you learn the reverse phase. This is the strategy. This is the trick how you have to learn. Because I know when it comes to exam, you all get confused. When such questions are there in the exam, it's quite obvious. I can totally relate with you that often we get confused, right? So the technique here is that if you're learning such concepts, learn one thing. You learn about normal phase or reverse phase. So if you're automatically learning about normal phase, you can easily answer reverse phase also, right? But yes, you have to understand the difference. That surely you have to understand. Very good. I'm so glad to see the answers. Mondru, Bhaskar, Anushri, Karthik, very good. Parveen, Chitra, Vidyariya, very good. Yes, you all are right. I'm glad to see your answers. So that means you're paying attention. Very good. And you're preparing well for the exam also. So keep it up. Very good. So I'll be giving you such concept checks questions in between. So be attentive, okay? So moving on to another chromatography, that is ion exchange chromatography. Now just see the term ion exchange. What is ion? I'm sure you have heard about two ions, cations and anions. Cations positive charge, anions negative charge, right? So we are exchanging with what? So let's find out. Ion exchange chromatography, it basically relies on attraction between oppositively charged stationary phase known as ion exchanger and analyte. So because as we are dealing with here charged particles, positively charged, negatively charged, so definitely here it has been related to attraction between oppositively charged particles. So one will be attached to an ion exchanger and another particle will be in your analyte, the substance that you want to separate. And mostly this ion exchange chromatography, it is being used for separation and purification of your proteins, peptides, nucleic acids, polynucleotides, and charged molecules. So you have to understand when you have to use a particular chromatography technique. It's not that for you can use any chromatography. It all depends on your aim of study. You have to use a particular chromatography technique. So what are ion exchangers? I have used the term here ion exchanger. So what it is? There are two types. One is cation, another is anion. Now, please note that here, like how you're going to understand, just see cation exchanger. So that means there is a matrix which should be negatively charged and it should attract positively charged cations. Okay. So always remember, now again here you have to learn one thing. Either you learn cation or you learn anion. Other, if you try to learn both, you will get confused. So cation exchanger possess negatively charged groups and they will attract positively charged cations. Anion exchanger have positively charged groups and they will attract negatively charged anions. Okay, so you can learn it this way: anion exchanger, so it will attract the negatively charged anions only. Cation exchanger, it will attract positively charged cation only. So in that case, matrix will be just of opposite charge. Okay, so here in this diagram you can see, so we have a beads. We use a polymer bead, right? So this is a column where we fill the beads. Okay, and these beads, they have a negatively charged functional group. So here I'm talking about the negatively charged functional group, that is, I'm talking about cation exchanger here. So you can see these beads have negatively charged groups, and let's say this is my mixture which has the components I want to separate. So what I am separating here, proteins. Now you know proteins, building blocks of proteins are amino acids, right? So the net charge on the protein can be positive charge, negative charge, depending upon what amino acids are there in the protein, right? So what will happen when you are adding the mixture? What will happen? The cation exchanger, the proteins with the more negative net charge move faster and elute earlier. Why? Because we have negative charge beads. So here, excuse me, please, this will attract positively charged cations, right? So just see the dots, like here these dots, they represents the different charge proteins. So you can see that the negative charge proteins are eluting first, and those with which are having the positive charge. So because the negatively charged matrix, the matrix is having negative charge group, they will attract positively charged cation, so they will be eluted later. Excuse me, please. So we can understand it in a form of a chemical reaction, that this is our anion exchanger, right? So let's consider an example of an ion exchanger. So an ion exchanger will have a positive charge group in the matrix, and to that negative charge anions are attached, and these are the anions which are present in the mixture. So these anions will be, this B1 will be exchanging the A1. So this is how the separation will be taking place. So here the charge is important, right? And even there are certain proteins which have both acidic and basic groups, right? So in that case, the ion exchange chromatography, it's pH dependent. So pH is also playing a very important role here. Now, you don't have to simply understand the principle of the technique, but you have to even learn that what are the examples of cation or anion exchangers used. Like for example, specifically focus on DEAE. So DEAE, you can see it's an example of an ion exchanger. Then carboxy or carboxymethyl, they are example of cation exchanger, right? Strongly cation exchanger are like strong acidic cation exchangers are sulfomethyl. So actually cation exchangers, you also call them as acidic, right? And anion exchangers, they are basic. So this way also you can learn. And why I'm asking you to learn the examples, not entire table, you don't have to learn the entire table, few examples you can learn. Because specifically I mentioned DEAE, you have to learn. Carboxymethyl you have to learn. Aminoethyl you have to learn. Carboxy you have to learn. Because again we can get a question directly on what type of anion exchangers are used, like what they prefer binding with. Or they can simply ask you that DEAE is what kind of exchanger? Is it cation exchanger or anion exchanger? Right? So can I get a answer quickly for this question? Diethyl aminoethyl, DEAE cellulose is what? It's an anion exchanger, right? So it will prefer binding with proteins having. Yes, quickly answer this question, everyone. Anushri, so quick, very good. Nishant, negative charge, very good. What about others? I can see some more participants. Let's answer this question, right? This is how we learn. When we interact, when we discuss, we learn more, right? Very good, Mondru. Vidya, Manvi, Karthik, Kartika, very good. Bhaskar, very good. You all have answered correctly. Answer 3, that is negative charge. Keep it up. Then next, moving to size exclusion chromatography. How many of you have heard about size exclusion chromatography? Can you tell me what are the other names with which we know this chromatography? Yes? Anyone? Parveen, yes, you are right. And Jason, yes, anion in chemistry, it's a negative charge particle. Sodium ion, cation, chloride ion, anion, right? Negative charge particle, Jason. Yes, can somebody please tell me that if I'm talking about size exclusion chromatography, what is the another name we know this chromatography? What could be the another name? Have you heard about it? Another name for this chromatography. Have you heard about gel filtration? Have you heard about molecular sieve chromatography? So are these chromatography same or different? Size exclusion chromatography, gel filtration chromatography, molecular sieve chromatography, is it same or is it different? Okay, Vidya, you don't know about it? Fine. So this is something new that you have learned today, right? That size exclusion chromatography is also called as gel filtration chromatography or molecular sieve chromatography. Okay, these are the name for the same thing. Fine. Parveen, very good. Yes, gel filtration, very good. So this chromatography technique is for the separation of molecule on basis of molecular size and shape. So when your aim of study is that you have to separate the molecules on basis of their molecular size and shape, in that case you will use particularly size exclusion chromatography. If you have the molecules in the mixture which are charged, then in that case you will use ion exchange chromatography. So always remember the keyword here is size and shape. Okay, for every chromatography you can make your own keywords. So for size exclusion it is size and shape. So here you can see that the stationary phase is actually, it's a column of microparticulate cross-linked copolymers which has a narrow range of pore sizes, and they are in equilibrium with a suitable mobile phase for the analytes to be separated. So here these are the beads which can be made up of the hydrated sponge like material which has pores. So here you can use the material like dextrin or agarose or polyacrylamide to make the beads, and you'll fill the beads in the column, and then you'll add your sample mixture. So if your sample contains large analyte, okay, these larger dots represents large analyte, the small dots represent small analytes. So larger analyte, what will happen? They are larger, so they won't be able to, they will be excluded from the pores, and simply they pass from the interstitial spaces which are present, right? Like here you can see they will be passing through the interstitial spaces. But when we talk about small analytes, they can enter the cavities, the pores, and therefore they will migrate slowly. So, what will happen when you're performing the size exclusion chromatography? The larger analytes will be excluded, will be eluted first, and then the smaller molecules will be eluted later. So, this is the point you have to keep in mind. Okay, so even I can show you how the separation is taken. You can even collect the the molecules which are separated. So here you can see this is the gel bead, and these are the red dots. It represents very small analyte, and the blue ones represents the larger analyte. So you can see larger analyte, they are just passing through interstitial spaces. They are excluded from the pores, and they're excluded from the pores, so they will be they will be eluting first, and then you have the smaller molecules, they will be eluting later. And what are the materials with which are the gel filtration material that you can use here? So we have the dextrin, we have a polyacrylamide, and we have an agarose. Okay, so here the beads, they are made up of these polymers of dextrin, right? So what is dextrin? It is actually, it's a polymer of what? Who will tell me? Dextran is a polymer of, even you have to understand what is dextran, what is agarose, what is polyacrylamide. Yes, and these are the trade names. So these are these beads are available with the trade names, like they are the products of the companies like G Healthcare. Cepadex is a product of G Healthcare. Biogel is a product of BioRed Laboratories. Yes, so what is dextran? It's a polymer of glucose. Okay, it's a polymer of glucose, and usually dextran, it has been used for separation of the smaller peptides, okay, or the globular proteins which have smaller average molecular mass. So even you have to understand when we are using dextran. So yes, Jason, Bhaskar, Shrivani, you all are right. It is a polymer of glucose. Okay, Jason, it's a polymer of glucose. Okay, and when you talk about agarose, so it's a linear polymer of alternating D-galactose and 3,6-N-hydro L-galactose, which has been isolated from red algae. And always remember, agarose, it has been used for the separation of large globular proteins, and even we use it for separation of DNA. And polyacrylamide, it has been formed by cross-linking acrylamide with NN methylene bisacrylamide, and it is also available in wide range of pore sizes. So if your aim is to separate smaller molecules, that is smaller peptides or globular proteins which are having smaller average molecular mass, you will use dextran. But for large globular proteins or for DNA, like in electrophoresis also we use agarose, right? Agarose gel. So for DNA you can use agarose. And even you can see how, like, these are the sizes, right? Their fractionation ranges. Then next, moving on to affinity chromatography. Shrivani, yes, you are right. Acrylamide, so polyacrylamide is being made by cross-linking acrylamide with NN methylene base acrylamide. Okay. Now, next is affinity chromatography. Affinity means something related to attraction. Now, here attraction, affinity means attraction. So, attraction of what? Attraction of a ligand with a receptor. Now, here you can see the two terms ligand and receptor. So, what is a ligand? Ligand is any group, any molecule which can attach to a macromolecule. Okay. So in this case, here in this particular diagram, I have again taken an example of proteins because chromatography is widely being used in like in R&D, especially for separation of the proteins. So here we have a mixture of proteins, right? So what you're going to do is that you will prepare a column where you will be adding the, like, you will be adding the column, in the column you will be adding a polymer bound ligand specific for protein of interest. So stationary phase, it is made up of a polymer to which the ligands will be attached. So what will happen when you add the mixture of protein, the protein specific to the ligands will bind. So that means to use this chromatography technique, you should have an idea, you should have a knowledge about the structure of your protein of interest you want to separate. So you should have a knowledge about the structure of the your protein of interest or your analyte of interest that you want to separate. Okay, so this is a preliminary knowledge you should have before performing this chromatography. So here you can see that to the ligand, these red ones are specifically binding and other ones are eluted. So these red ones are our protein of interest, right? And finally, once unwanted proteins are washed off through the column, you can add another solution to separate the protein of interest, right? So here the beads in the column that we have used, they have a chemical group, right? So ligand, it's a group which has been, it's a group which can attach to a macromolecule. So here your macromolecule is the protein, right? So like for example, if you are separating fatty acid based, like the, like the one example of ligand in receptor is that you have a ligand fatty acid. So fatty acid binding proteins you can separate. So you can use fatty acids as a ligand, and fatty acids binding protein will be, you will be adding, okay, the mixture that will be in the mixture. So you can separate fatty acid binding protein by performing affinity chromatography. Now lastly, coming to gas chromatography and high performance liquid chromatography. So here I've added a comparative table because it's very important that you understand these two chromatography techniques thoroughly. Okay, why? Because both the techniques are very important, and when you compare you learn better, right? So this is the reason we use this. I have used a comparative approach here. And Parveen, R&D, research and development. Okay, in research and development in the lab, in the industries, in the laboratories. Okay. Any other question? Please post in the chat. Okay. Meanwhile, I'll explain you what is the difference between gas chromatography and high performance liquid chromatography. So just see the diagram. You can see in gas chromatography system we have a oven, and we have a we have the column which is coiled, right? And here why we are calling it as gas chromatography. So if you remember, I told you, if when you are using a mobile phase gas, then it is a gas chromatography. If you are using a mobile phase liquid, then it is liquid chromatography. But why we are calling it as high performance liquid chromatography? Because here we are using high pressure pumps, okay, and they provide better resolution. It's better separation of the components in a mixture. So high pressure pump which can withstand 50 megapascal of the pressure. Then we have ultra performance liquid chromatography also, which can withstand 150 megapascal pressure, right? So here you can see the difference. There's a difference in the column. There is a difference that in gas chromatography, oven is used, but it is not used in HPLC. The short form of high performance liquid chromatography is HPLC. And gas chromatography, you can write GC, right? And then both the chromatography systems have recorders and detectors, right? So let's compare both of them. So first you have to remember the principle on which these two chromatography work. So gas chromatography, here the separation is based on partition coefficient. Please learn this concept. And in HPLC, the separation can be based on adsorption, ion exchange, size exclusion, or reverse phase. RPC is reverse phase chromatography. I have used a short form here. Then sample, it has to be volatile. Volatile means the sample which can be easily converted into gas, because here you are using an oven, right? So when your sample is in the oven, your sample is in the column, so you are heating the column so that your 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 sample should be in the gas phase, right? So samples should be volatile, and usually low molecular weight samples we prefer gas chromatography. And yes, it should be stable. It should not get denatured because when you raise the temperature, your sample should not get denatured. So it should be stable. However, for HPLC, you can use volatile or non-volatile, low or high molecular weight sample. Your component can be that you want to separate can be volatile or non-volatile, low or high molecular weight. And usually in HPLC, we don't provide high temperature. It has been performed at room temperature only. But when you compare the column, so column can be long and narrow in gas chromatography. You can see it's very long. It is in coiled form here. And we have two type of columns: packed column, which is a conventional column, and another is a capillary column. So packed column is a conventional column which is made up of glass or stainless steel, and capillary column, it is the open tubular column, and it is made up of stainless. It is made up of high quality fused quartz. Okay, and they differ in terms of their length and diameter. For HPLC, the column is usually short and wide, as you can see here. Okay, it is short and it is wide. Just see here, it is wide, right? And it is a packed column, conventional column that we use here. Now, what is the difference in mobile phase? So in gas chromatography, it's an inert gas. You can use hydrogen, you can use nitrogen, right? And no pressure you have to provide here. And even the mobile phase you can use here, like no gradient is required. But in HPLC, solvent mixture is there. Okay, your mobile phase is a mixture of solvent, and you can use mixture, like gradient can be applied. See, we have two terms: isocratic and gradient. Isocratic is where a single pump is required. Like here you can see we have a pump to uh, to provide to pass the mobile phase into this column, right? To pump the mobile phase into the column. So actually the point is that in isocratic single pump is required, but in gradient two pumps are needed. And in isocratic only one eluent is required, but in gradient we use two. Okay, so isocratic one eluent means the solvent that will pass through the column, that is one only, but in gradient it will be two. So you are providing the mixture of the two solvents that will pass through to the column. And yes, pressure is required, that is why we are naming, I mentioned here, high pressure pump is required. Then even you can learn the detectors. So UV visible detectors are used in HPLC, refractive index detector, fluorescent detector, mass spectrometer detector in HPLC. So you don't need to go in detail, but just simply you can learn the name, because maybe directly a question can be asked on what type of detector is used in gas chromatography or HPLC. So here the table, you can easily go through it. The cost of gas chromatography, if you talk about the cost, then gas chromatography is comparatively low cost. HPLC, it is high cost. And even you can see the applications that we usually use gas chromatography for separation of pesticides, fatty acids, toxins, oils. But for HPLC we use it for separation of sugars, vitamins, peptides, proteins, lipids. Okay, so this table is very much important. And Parveen, what is capillary column? Capillary column, it's an open tubular column. Okay, it's an open tubular column, another name is open tubular column. Okay, and it is made up of high quality fused quartz, and it can be around 10 to 100 meter long. Fine. And they are around zero point 0.1 to 1 mm internal diameter they can have. So why column? See, why different type of columns? Because see, every technique has its own advantage and disadvantage. So maybe the conventional columns that we are using, see, at the end it's in the column where separation is taking place, right? So we have to improve the resolution. For that we have to increase the quality, we have to change the, we have to check, we have to test that if we change the diameter, if we keep the length, we keep the about certain dimension, if we keep the diameter of certain dimension, in what we are getting a better resolution, better separation, right? So scientists keeps on improving the techniques, right? They are keep on they are try to modify it so that they can overcome the limitations associated with the conventional technique, right? So I hope it is clear to you, Parveen, now what is capillary column, right? So last, yes, now finally coming to application of chromatography. So you can see here we have a lot of applications, right, of chromatography. We can have it in pharmaceutical sector, we can have it in chemical industry, food industry. So like for example, in pharmaceutical sector, we have like, you have a, you want to find out the amount of chemical which is present in a drug, in a product, right, drug product. So for that you can for here you can use chromatography in pharmaceutical sector. Then in chemical industry you can use it, right? Like if you are making any product, so you can analyze the components in it. Then food industry, like in the middle of the session I gave you an example that if you are testing the food quality, so there are several food coloring agents which are used, right? But that food coloring agents are only used which are being which only the agencies, the regulatory agencies permit. But nowadays, you know, there's a lot of adulteration and lot of other and these drugs that these agents that we are using, right, coloring agents, they should be used in a specific limit. If you overuse them, then they will definitely cause a health problem. So even in food industry, for analysis of these coloring agents, you can use, like TLC is widely used for this purpose, right? Other chromatography you can use. Then in forensic science, so in forensic science, let's say you find a sample at the crime scene, so from the suspect also you can take a sample and you can compare, you can analyze both the sample using chromatography. Then molecular biology study, widely we are using chromatography technique for separation of the proteins in the laboratories, right? So this is what the basic overview of what are the applications of chromatography. So you can see they are used in different areas, and this is the reason I told you this technique is important, because it has been widely used in different areas, different field of life sciences, right? So quickly let's answer a few questions. Separation of components in gas chromatography is based on what? It's based on molecular weight, is it based on partition coefficient, molarity, or conductivity? Yes. Can you quickly answer this question? It's the this question. If you learn that table, you can easily answer any question based on that table, right? So make sure that the notes will be provided in the end, right? You have to fill the feedback form. The notes will be provided. So make sure that you learn that table. Partition coefficient, very good. Nishant, Vidya, Manvi, Anushri, Chitra. I'm glad to see your answers. Azagya, Parveen, Ria, Jason, you're right. Shivani, you are saying molecular weight. Molecular weight, you remember? Size exclusion chromatography. Make a keyword for every chromatography. Yes. No, so here I'm talking about gas chromatography. So very first thing I mentioned here is that in gas chromatography, separation is based on partition coefficient, right? So here the answer will be 2. Mondru, you are right. Kartika, you are also right. Avishka, you are also right. Very good. Keep it up. Now, let me ask this question. We have a lot of easy questions. Let's have a little bit complicated one, which can be quite confusing. Here you can see the options are quite confusing. Just go through this question. The question is again about gas liquid chromatography, that there are a large number of stationary phases available, while in HPLC, high performance liquid chromatography, there are wide range of stationary phase, wide range of mobile phase, wide range of both stationary and mobile phase, or is it narrow range of stationary phase and wide range of mobile phase? What do you think? Yes, let me have the answer quickly. What should be the answer here? It is already mentioned that in gas liquid chromatography, there are large number of stationary phases available. But in HPLC, what do you think? Okay, Shrivani, you say option D. Okay, let's see what others are answering. Wide range of mobile phase. So you are saying Srivastav, Avishka Srivastav, you are saying option B. And Azagya, you are saying option D. Manvi, you are saying option D. So I think there's a confusion between B and D, right? Because some of you are answering option D, some of you are answering option B. So here I have a suggestion that see, you all have your own technique of studying, right? But whenever you are studying anything, whenever you are even solving questions, see, sometimes we learn the concepts when we solve the question. So this is the importance of solving questions. Sometimes we learn the concepts better when we solve the question. So don't simply do the rectification. Okay, we don't have to simply rote memorize that, okay, this is what happens in HPLC, this is what happens in GLC. Try to relate, try to compare and learn, or you have to understand the concept behind it. So here the concept, like what should be the answer for HPLC here? So always remember, as I say, when you compare, you learn better. So in gas liquid chromatography, we can get a better result when you are using a wide range of columns packed with different stationary phases. So it means that you are using large number of stationary phases. But always remember this line. In the case of HPLC, reverse is true. Reverse means that you keep the stationary phase limited and you vary the mobile phase composition, right? So you can keep the stationary phase limited. Either you can use the support of silica or alumina, right? But the composition of the mobile phase you can vary. And when it say that you can vary the mobile phase and the reverse is true, so that means the appropriate answer should be option D, right? So obviously in exam you will get some questions where the chances of confusion will be there. Here's the confusion is between B and D. So when we understand the when we compare, when we understand in such way that what kind of stationary phases are used in one type, what is the mobile phase used in both these type of chromatography, what is the difference, you will understand the concept better. you will retain that concept and you will never forget. So always remember, highlight, okay, just keep such points in mind. So here always remember, in HPLC reverse is true. So for this you have to just remember one point, that's in gas liquid chromatography, wide range of stationary, large number of stationary phases we can use. So for HPLC reverse is true. Only this line you have to keep in mind. So Jason and Shrutika and Mondru, is it clear to you why the answer will be option D here? Yes. Now let me quickly move on to spectroscopy. So this is another part of the session. I will not take much time. So let's quickly understand what is spectroscopy. So again, we have to understand what is the meaning of spectroscopy here. So spectroscopy, it is actually derived from two words. So spectrum, which means image in Latin, and scopia, which means observation in Greek. So spectroscopy, you can say it is a branch of science which deals with interaction of matter with light. Matter is what? Anything which has mass and occupies space. So it can be any biomolecule like DNA or protein, right? So here you study what happens when the light, it interacts with the matter. Whether, so when light interacts with matter means several phenomena happens, right? Either the light will be absorbed, it will be transmitted, it will be scattered, right? So this is what we study in spectroscopy, and it's a both qualitative and quantitative technique. That is, it will not only help us in analyzing the qualitative, but quantity also, quantitative. Like for example, if you are analyzing DNA, so where does DNA come from the cell. So you have to isolate the DNA, but it is not the DNA only present in the cell. You have the proteins also, you have RNA also, right? So when you are isolating the DNA, when you have to use the DNA for further analysis, you have to make sure that that DNA should be free from contamination. So here the contamination is anything other than DNA is your contamination. It can be RNA, it can be protein, right? So you can check the quality, you can check whether your DNA is pure or not by using spectroscopy, and even you can check the concentration. You can find out how much is the concentration of the DNA present. That is why I'm saying it's both qualitative and quantitative technique. And it became a very scientific tool in 19th century with the spectrochemical analysis of material composition, which was established by the German scientists Gustav Kirchhoff and Robert Bunsen. Now here I have used the term light, okay? Like I have used a term that interaction of matter with light or electromagnetic radiation, right? So what is light? Parveen, I'll come to that point, that how we check the quality, okay? You have to wait for a minute. I'll explain you everything, don't worry. So I'll come to the quality. Let me just, I'll talk about qualitative analysis in detail later. Let me first tell you what is spectroscopy and its basics. So here I have used the term light. So light is what? It is nothing but an electromagnetic radiation which provide a sense of sight. Like if I ask you to enter into a dark room, will you able to see anything? No, right? You need light in that room to see the furniture in the room, right? To see where is the gate, where is the window. If everything is closed, if there is a lot of darkness in the room, you are not able to see anything. So light is an electromagnetic radiation that provide a sense of sight. And what is electromagnetic radiation now? Electromagnetic radiation is a form of energy, right? Which transfers in a form of a wave, right? And so light travels in a form of a wave, and this electromagnetic radiation has a discrete packages of energy which we called as photons. So these photons, they have an oscillating electric field and magnetic field perpendicular to direction of each other. This is the reason we are calling it as electromagnetic wave. There is a reason for everything. Okay, so why we are calling it as electromagnetic wave? Because here the electromagnetic radiation consists of discrete packages of energy called as photons, which have electric field and magnetic field oscillating, like they are perpendicular, oscillating electric field and magnetic field perpendicular to the direction of each other. And this electromagnetic radiation can have numerous forms. And that forms the electromagnetic spectrum, which you all can see here. So again, dear participants, please learn this particular diagram. It's very, very important from exam point of view. This diagram itself is a summary of different type of spectroscopy techniques that we use. So here you can see that we have X-rays, we have UV visible ray, we have infrared, we have microwave, we have radio wave. So based on what electromagnetic radiation you are using, we have named the spectroscopy technique accordingly. Like you might have heard about infrared spectroscopy, Raman spectroscopy, microwave spectroscopy, radio. You have studied about NMR, nuclear magnetic resonance, right? So why these names? Because specific type of radiations are being used here. So these radiations that I'm talking about, they have a specific wavelength, they have a specific energy, they have a specific frequency, right? So these are all the wave properties: wave number, frequency, wavelength. And you can learn the ranges, you can learn the wavelength of these radiations either in angstrom or in nanometer. Okay, nanometer you can learn. Like for visible region, we say the range is 400 to 700 nm, right? Now question arises, when to use which radiation? So it all depends on your aim of study. If you want to study changes in electronic state, please note you will be using X-ray or UV visible radiation. If you want to study rotational and vibrational state, you will use infrared or Raman. If you want to study changes in only rotational state, you will be using microwave. And even in ESR, we use microwave. Okay, if you want to study electron spin, then also you use microwave. If you want to study nuclear spin, you will use radio wave, and the spectroscopy name is nuclear magnetic resonance. Right? So this particular diagram itself is very, very important. So let's find out what happens when the light interacts, electromagnetic radiation interacts with matter. So matter, the basic unit of matter is atom, right? Now this atom has a central nucleus and electron cloud, like there are 3 subatomic particles. I'm sure you all know about it, right? We have protons, neutrons, and electrons. Protons and neutrons are present in the nucleus, and electrons are present outside the nucleus in different orbitals. So what happens when the light of specific energy or wavelength, it is provided, electromagnetic radiation of specific wavelength or energy, it falls on the matter, a biomolecule, the atoms, the electrons present in the atoms, they absorb the energy and they jump from one energy level to another. So here you can see this diagram. The energy level on which the electrons are present, we call them as ground state. And on absorbing the radiation, they move on to higher energy level, which is excited state. Okay, so excited state is temporary. It's higher energy state. And everything in nature has a tendency to acquire a lower energy state. That is, the electron will again come back to ground state. So when you are studying the absorption of the light, the spectra which is obtained by absorption of electromagnetic radiation, that is called as absorption spectra. And the spectroscopy that we use to study absorption spectra is called as absorption spectroscopy. So example is UV visible, infrared, right? These are an example of absorption spectroscopy. However, when you are studying about the emission spectra, so emission of light, how it is obtained? This electron will again come back to ground state, and in doing so, it will releases the photons of light. So the spectra which is obtained by emission of electromagnetic radiation, that is emission spectrum. Okay. And like fluorescence spectroscopy is a type of emission spectroscopy. So broadly, we have divided the spectroscopy in two parts, absorption and emission. And here, please note everyone, that here this h mu is nothing but this is how we represent energy, that h is equals to Planck's constant, h is the Planck's constant, and mu is the frequency. So definitely this energy difference, how you can calculate? Delta E. So delta E is how much? It's the energy of excited state minus energy of ground state. So that means if the electron has to undergo transition from ground to excited state, that is E1 to E2, it has to absorb this much amount of energy, delta E. Then only it will undergo transition, right? So here I have a table for you all again. So you can see these are the different electromagnetic radiations that can be used. So these are all absorption, these are the radiations that you can use to study absorption spectra. And based on what radiation you are using, you have named the spectroscopic technique accordingly. And you will use a particular radiation depending upon what you are studying. So if you are studying the electrons, right, if you are studying the outermost electrons, valence electrons, right, they transition from one energy level to another, one energy state to another, then you will use ultraviolet, right? If you want to study rotational and vibration state, you will use infrared. If you want to study only rotational state, you will use microwave, right? Radio wave you will use to study the nuclear spin. Similarly, emission. So here you will be studying about emission spectra. So here I would like to show you, okay, this is called as Jablonski diagram. Okay, in molecular biology, we use this diagram to depict different energy levels. So here you can see every electronic energy level has its own vibrational and rotational energy level, right? So if the transition has to take place between vibrational states, you can see it's very less energy is there, very less energy difference is there. But for transition of the electron from vibrational state of one electronic energy level to vibrational state of another electronic energy level, definitely this much amount of energy has to be, delta E has to be absorbed, right? So these points you have to keep in mind, that what you are actually studying, based on that you will be using a particular radiation, right? Like you may ask that, ma'am, why we are using UV visible only to study the electron, why we are not using infrared for that? So here you can see that the energy difference between electronic energy level is more, right? And as I mentioned earlier, that be it UV visible, be it infrared, they have a specific wavelength, they have a specific energy, right? So obviously you have to provide the radiation which has that much energy, which can provide which can cause the transition of electron from one energy state to another. So UV visible region has this much energy that can provide this transition to, that can cause this transition to take place. Okay, and one more point here you can remember, that relation between energy and wavelength is inversely proportional. You can learn this keyword HHS. Okay, the relation between the wavelength, the frequency, and the energy is HHS. Higher the energy, higher the frequency, shorter will be the wavelength. So X-rays, UV visible, they have very high energy in comparison to radio wave and microwave. So higher energy means shorter wavelength. Excuse me, please. Now coming to kind of transitions. Always remember, like if I give you an example of UV visible spectroscopy, dear participants, I will take 10-15 minutes more. I hope it is fine with you all. Let me just complete with the spectroscopy. I'm sure you are enjoying the class. Yes, you are learning some new concepts. Will it be fine if I just extend for 10-12 minutes? Please answer in the chat. Okay, thank you so much. Thank you so much, Avishka. Really means a lot. Your response really means a lot. Thank you, Nishant. I'm glad to see your responses. Thank you so much. And I hope the others are also fine with it. Oh, thank you, Vidya. Thank you, Chitra. Parveen. Thank you so much. So, yes, now I'll be telling you some very important points. Please pay attention. The electron transition will depend upon, like the absorption of light will depend upon the type of electrons which are present. So we have three type of electrons. One is called as sigma electrons, which are present in saturated compounds, that is those which don't have double bond or triple bond. Pi electrons, which are present in unsaturated compounds, like which have double bond or which have triple bond, right? And non-bonding electrons, which are present in like nitrogen, oxygen, lone pairs, where they are present, right? So again, this diagram you have to learn. So it's an energy scheme for molecular orbital. So here I'm telling you about the absorption spectra. Now one more point. What is spectra? Spectra is actually a graph where you can plot the absorbance by wavelength or frequency. This is how we get the result of spectroscopy. Okay, so spectra means it's a graph plotted where on one axis you have the absorption or emission, and on another axis wavelength or frequency. So you can measure at what wavelength how much absorbance is taking place. And that for every macromolecule that you are studying, like DNA or protein, they absorb at a particular wavelength, right? And we get the result in form of a peak. In modern spectrophotometers, they plot a data for us, so in the form of a peak, where the peak height of the peak is proportional to the concentration. More larger the peak means more the concentration, right? So here you can, coming back to the transition, so here you can see that transition is from pi to pi antibonding, right? Non-bonding to pi antibonding, non-bonding to sigma antibonding, and sigma to sigma antibonding. So these 4 transitions you have to keep in mind. So the black arrows are actually representing the transitions which are possible when you provide UV visible radiation. Thank you, Shrivani. I'm glad you are enjoying the class. Thank you so much for your response. And the gray arrows here, they are actually representing the transitions which are not which are not in UV visible range. Okay, you need more energy. So like near the X-ray range, you need X radiation you need. So this is very important concept you have to remember. Okay. So what is bonding and anti-bonding? Let's say there's a hydrogen molecule. So how hydrogen molecule will form? The atomic orbitals of these hydrogen atoms, they will overlap and they form 2 molecular orbitals, right? So if the attractive force, like when the electrons are infilled in the orbitals, so the attractive forces in the nuclei, if it is it is allowing these or this orbit this like stable interaction to take place, then it is a bonding orbital. It is lower in energy. But higher energy, it is always anti-bonding. Okay. So this is the point that you all have to keep in mind. Okay. And if you have any other question, you can post right now. Fine. So here, this is the energy scheme of molecular orbitals that you have to remember. And another very important topic that you have to remember that is associated with spectroscopy is Beer-Lambert's law. Fine. So bonding and anti-bonding, I hope it is clear. So one which will favor the nuclei to be overlapping, right? The atomic orbitals to overlap. So one orbital which is lower in energy is bonding, and one which is formed higher in energy is anti-bonding. And usually the electrons, they prefer to first occupy the bonding orbitals. And if the transition has to take place, that means you can see so much difference. This much amount of energy has been required for transition of electron to take place from bonding to anti-bonding, right? Then absorption spectroscopy, if I specifically talk about, they all follow Beer-Lambert's law. So it's a combination of the laws, one given by Beer and another by Lambert. So it's called as Beer-Lambert's law. So according to this law, the amount of light which is absorbed by the solution or the sample at given wavelength is always proportional to concentration of absorbing material or solute present. And so this is Beer law. And Lambert law states that amount of light absorbed by medium is proportional to thickness of absorbing layer or path length of the light. So here in spectroscopy, we are studying about the interaction of light with the matter, right? So let's say this is a tube, we call it as CUET, where we are placing a sample, right? Let's say it is a DNA. So according to Beer-Lambert's law, as I mentioned earlier, that when the light it is falling on any substance, several phenomena can take place. Either the light will be absorbed, it can be transmitted, or it can be scattered, right? So here, when the light falls on your macromolecule DNA, the light absorbed will be directly proportional to the concentration of the DNA present. Okay, concentration of the absorbing material or solute present. So more the DNA, more will be light absorbed. And always remember, there is an inverse relationship between absorbance and transmittance. That is, if more the light absorbed, less will be transmitted, or vice versa. And even the amount of light absorbed will be proportional to this thickness of absorbing layer. So this is the path length. Okay, the path which is being, this is the path length, okay, the thickness of the absorbing layer which is there. So let's suppose I0 is the intensity of incident light and I1 is the intensity of transmitted light. So according to this Beer-Lambert's law, absorbance is always, how you can plot the ratio for transmittance and absorbance. So for transmittance, it is ratio is transmitted intensity by incident intensity, and for absorbance, it is always measured in logarithmic scale. So it is inverse, right? Absorbance is inversely proportional to transmittance. So it will be incident intensity upon transmitted intensity. So if you are writing it like this, minus log transmitted intensity upon incident intensity. So if you have to remove the minus sign, you will write log incident intensity upon transmitted intensity. This is one way of expressing the Beer-Lambert's law, and another is that you can write down absorbance is equals to molar extension coefficient, concentration, and path length. So C is the concentration, L is the path length, and this is epsilon. This is actually the molar extension coefficient, which is the property of any compound. Okay, what is molar extension coefficient? It indicates how readily the compound will absorb the light. So obviously it will depends upon the structure of the compound, the chemical species, right? So this is molar extension coefficient, and for every chemical species there is a molar extension coefficient value. You can calculate the absorbance, you can calculate the concentration, right? So if you know the molar extension coefficient, if you know concentration path length, you can find out absorbance. Similarly, if you know absorbance, if you know molar extension coefficient, if you know path length, you can find out concentration by simply rearranging this formula, that absorbance by molar extension coefficient and path length, right? So this is how we express the molar extension. This is how we express the Beer-Lambert's law. And now let me quickly tell you how you will measure the concentration. Like we're talking about measuring the concentration, right? Which is directly proportional, or we are measuring the absorbance. You are finding the absorbance, which is directly proportional to the concentration. So here, this is the instrumentation which I have shown for UV visible spectrophotometer. So here the obviously we'll be needing the lamp, right? So for visible light, you can use tungsten filament bulb. For deuterium bulb, you can use ultraviolet. Deuterium bulb you can use for UV region. Now always remember, the light which is coming out from these bulbs, they are polychromatic, that is of different wavelength. But for analysis, we have to use monochromatic light, that is light of single wavelength. So to convert the polychromatic into monochromatic light, you have to use a monochromator. Okay, so monochromator, either it can be a prism you can use, or grating, rotating metal grid of higher precision, we call it as grating, you can use, right? So what will happen when the light will pass through prism? It will disperse into different colors. Like if you talking about white light, it is made up of, it has 7 colors, so it get dispersed into 7 colors, right? So light of specific wavelength, you will, with the arrangement of the slits and mirror, you will allow it to reach to the CUET where you have the sample, and finally you will measure the absorbance. So detector will detect the light absorbed, and the data will be available on the screen. You can use reference CUET also. So this is a kind of a dual beam spectrophotometer. So reference CUET is like, for example, if if you are analyzing DNA, so obviously you have to suspend the DNA in some buffer, right? TE buffer, TCDTA buffer. Now this buffer has some ions which can absorb the light. So in that case, you will get a false result. You will not get the actual absorbance. So this is the reason you put the solvent which does not carry the DNA, the solute, and you can take the absorbance. So let's say you get some absorbance. So you can omit that background absorbance from the actual absorbance of your solute or your sample. Right? So what are the applications? Why we use spectroscopy? Okay, Shrutika, you asked me that molar extension coefficient is of DNA or solvent use. Molar extension coefficient of every chemical species that you are studying. So that chemical species, whatever you are studying, it can be organic compound, it can be DNA, it can be protein. So in proteins, you know the absorbance is because of amino acids, aromatic amino acids, right? So tryptophan has a molar extension coefficient, phenylalanine has a molar extension coefficient. Clear to all? Chemical species which are present. So how readily they can absorb the light, it all depends upon the, like if any structure has a pi electron, so you can see that pi to pi transition can take, pi to pi antibonding is taking place very easily. Like benzene ring, it has a conjugated double bonds, right? So pi to pi electrons are there. So any very less amount of energy is required for this transition to take place, pi to pi antibonding. Similarly, non-bonding to pi antibonding transition can occur very readily. But for transition from sigma to sigma antibonding, it requires lot of energy. Clear to you now, Shrutika? So what is the application of spectroscopy? So you can use it in we use spectroscopy in food, right? Food industry for measurement of different compounds which can be present in food samples. Then in academics, we widely use it to determine the concentration of your DNA, protein, RNA, to check the quality, to check the quantity, right? And one of you asked me that, ma'am, how we can measure the quality and quantity. So I would like to explain that point in detail. So here you can see that always, whatever biomolecule we are studying, they always have a lambda max. That is the wavelength at which maximum absorption will take place. Usually it's a range, right? Like nucleic acid, they have a range where they show absorbance. But lambda max is the wavelength at which the maximum absorption will take place. So please note, for nucleic acid, it is 260 nm. For proteins, it is 280 nm. And even we have standard values, like if you are measuring the concentration of nucleic acid and you get the absorbance 1 under standard condition, so they corresponds to 50 microgram per ml for double stranded DNA, 33 microgram per ml for single stranded DNA, and 40 microgram per ml for RNA. So if you get absorbance 2, that means, if you know the standard values, you can multiply by 2. So that means the concentration of nucleic acid will be 100 microgram per ml. So this is the importance of these standard values. You can determine the concentration of your sample, right? And then how we can check the quality? So for checking the quality, we have to take a ratio. Like you will be measuring the absorbance at 260 and also at 280. And if you get the ratio 260 by 280 around 1.8 to 2, okay, that means your sample is pure. Your DNA sample is pure. But if you are getting the absorbance less than 1.8, it indicates protein contamination. And if it is more than 2, then it indicates RNA. So, Parveen, I hope this answers your question, that how we can check the quality of DNA. Is it clear to you? You will be taking the ratio. Similarly, if you take the absorbance at 270 nm, if you get a P, that means it indicates phenol contamination. Right? So coming back to the applications again, you can use it in clinical spectroscopy. You can use it in clinical studies also, right? And pharmaceutical analysis. So like pharmaceutical analysis, you can use spectroscopy for elemental analysis. You can study the structure of the drugs, so that you can develop improved drugs. You can improve them for their effectiveness. Then in environmental analysis, you can use spectroscopy to measure the dissolved oxygen content in freshwater and marine ecosystems. Astronomy also, it has a very important role. You can study spectral emission lines of sun and distant galaxies. Then in forensics, you can study the toxic compounds which you can find in blood samples collected from the crime scene. So here you can see there is a lot of applications of spectroscopy, and it's a very, very important technique. And I really congratulate all the participants who have attended this session, because today you have learned a very, very important, you have studied about a very important technique. And you will actually understand the value of this session when you will actually be performing these techniques, performing the experiments about these techniques in the laboratory. Because see, unless and until, I'm sure you'll all agree that you will understand the technique better when you perform it practically. But to perform a technique in the lab practically, you have to first know, you should need to know about the theory, why you use that technique, what that technique is all about. If you don't know the basic, then then you will not think about its practical aspect, right? So thank you everyone for attending this session. And let me just quickly discuss few questions based on this spectroscopy also. So the questions that can be asked from this particular topic is, like, according to Beer-Lambert's law, what do you think? What is absorbance? What absorbance is? Quickly answer this question. According to Beer-Lambert's law, absorbance is what? Shrivani, very quick, very good. Directly proportional to concentration. Simply have to remember this formula. Absorbance equals to directly proportional to concentration. Right? Very good. You all have answered correctly. Then another type of question is UV visible spectroscopy of organic compounds generally concerned with which electronic transition? So you remember black arrows specifically I mentioned. So this question is based on that. So again, can you tell me what kind of transitions, electronic transitions are possible here in UV visible spectroscopy? It is concerned with basically which electronic transitions? See, any option having sigma to sigma anti-bonding, you can omit, right? Because it requires very high energy. I told you near to the X-ray um, range, right? The X-ray radiations, that much energy we need. So obviously, first two cannot be the answer. So that means what is the answer? It should have pi electrons or non-bonding electrons. So answer here will be option 3. Okay, answer here will be option 3. So out of 4 possible transition, this one is pi. Okay, only 2 can be elicited with light with UV visible spectrum, right? So these are the 2. So that means answer is option 3. And non-bonding to sigma anti-bonding or sigma to sigma anti-bonding transitions are energetically not within UV visible spectroscopy and require higher energies. And the last question is that in order to check the purity of DNA sample, ratio of absorbance at 260 by 280 nm is measured. For a pure DNA sample, the value of absorbance 260 by 280 is? Yes? Okay, optimistic mind. Option 1, Chitra, Nishant, Anushri, Manvi. Okay, you are saying option 1. So it is 1.8. Okay, 1.8. Yes, it's 1.8. Okay, it's answer 1. 1.8. So, with this, I have come to the end of this session. I'm really glad that you all have attended this session, and I'm really glad to see all your responses. It was a really wonderful interactive session, and I request my team to just in the chat they will provide you the feedback link, and you will also get the notes for this session. So, I'm sure you have enjoyed the session. I can see from your responses, and so I'm sure you'll be attending the next session as well. So, thank you everyone and all the very best with your preparation. Remember, BioTecNika is always with you. You are not alone in this journey of preparation for CUET PG exam. We are always there with you. Thank you, everyone.